Good evening everyone. This is your ninth week session with me, and in today's session, we'll be looking into week 9 topics in which we'll see the applications of genome editing in treating the human diseases, in which we will see animal models, why they are used, and what are the different types of animal models, what are different advantages of using them. We will see iPSC models and different cancer disease models. Then we will practice few MCQs, we'll look into the current literature, and then I'll be taking your doubts. So first of all, the foremost thing is that let's understand that what is the importance of these disease models. So first of all, the main advantage is that these are used to replicate the human disease processes in the animals or cells. For example, if I want to know about a disease, so instead of studying it in a human, we can study it in the animals and other small cells, so that we can know that what could be the better therapeutic interventions that could be made, what are the disease progression stages. All those things can be replicated, we can study them, and it is used to study disease mechanisms, their progression, their prevention and therapy. It is essential for drug discovery, toxicology and regenerative medicines. So we can discover certain drugs. So for example, there are certain cell lines or E. coli, certain prokaryotic cells, bacterial cells. So in them we can study that what is the effect of those drugs instead of directly giving them to the human host. So those things can be done. Toxicology can be analyzed and regenerative medicines can be generated. Models reduce the ethical and technical barriers of direct human studies. So there are certain barriers due to which if we use the model organisms, then it reduces those barriers. So that leads to the translational research. So which means that we have different model organisms such as zebrafish. So there are certain advantages of each of these model organisms. So if we start with the zebrafish, then it has transparent embryos. That means whatever we are doing with its embryos, we can directly see them because they are transparent. Then genetic manipulability. So we can manipulate their genome. We can make the changes. If we want to study certain disease and if it is related to particular gene, because these organisms share some kind of gene similarity with us, so we can specifically target that gene, we can mutate it, we can modify it to see that how it leads to the change in phenotype in that particular organism. So in that way it is known as genetic manipulability, that we can manipulate the genes. Rapid external development, so they shows rapid development, so we can study the effect quickly. Conserved genetic pathways, so they have certain pathways which are conserved across them also and us also, so we can study them, that how the different pathways got affected. Behavioral assays, we can study their behavior. And regenerative abilities, so the zebrafish has the ability because it can regenerate its eye and different organs, so we can study that what are the genes which are responsible for those regenerative abilities, so that if we could understand that thing, so we can apply that to human as well. Another organism is Drosophila. So actually I just started with zebrafish, but I should have ideally started with Drosophila, because Drosophila is an invertebrate. So Drosophila it has genetic tractability. We can track its genome. It has short lifespan, so we can quickly study certain things because it has short lifespan. So the study can be done quickly. It has simple nervous system. It has high reproducibility. Behavioral assays can be studied, and it is cost effective and maintenance is quite easy. Then if we talk about Xenopus, which is this frog, so it has large embryos. It has optical clarity, rapid development, regenerative potential, genetic similarities and easy maintenance. So these are some of the features of the frog. Then comes rodents. So genetic similarity to humans, complex nervous system, they have behavioral complexity, disease modeling can be done, well established genetic tools are available with which we can study them. Behavioral assays can be done. So behavioral assays means that we are observing the behavior, that how the behavior gets changed with influence of certain chemicals, certain genetic modifications, perturbations. So all those things can be done with respect to the clinical research. So this kind of research is translational research, where we are translating something from these organisms and using it for the human. So we are doing translation kind of thing. Now, what are animal disease models? So they are defined as known human species. When we say animal disease models, although human is also under the kingdom Animalia, but disease models we only use known human species which mimics the aspects of human disease. So we will only use those species which can mimic the human disease effects. We cannot use those animals which are completely away from human. So according to NIH, animal models share the physiology, anatomy and disease response with humans, and they are used for drug testing, in which we can check for the safety and efficacy of the drug, that whether it's safe to consume, whether it's effective or not. Understanding the disease biology, we can understand that how certain disease get progressed, what is the biology behind that disease, what are the genes responsible for it, what are the pathways which get affected, and means whatever, we can study the entire biology related to that disease. Testing the surgical and regenerative procedures. So examples of animal models are mice, rats, zebrafish, rabbits, pigs and primates. So these are the known mammalian models such as zebrafish, flies and C. elegans, which is nematode. So they have low maintenance cost because they are less complex organisms. Then comes rodent models. So they are cost effective, mouse, rats, and they also share high similarity with respect to the human genome. So they lack, means the rodents are more closer to human as compared to known mammalian models. Then comes larger animal models. So they have relatively high cost. Now assume that you are doing some study on Drosophila. How much nutrition, how much care do you need to? The place where you are keeping this will be very small. The nutrition which it requires will be very small. So means maintenance is very low and it is cheap. Whereas if I'm keeping a rabbit, I'm doing studies on it, it requires a greater space, it requires greater nutrition, more care need to be taken off. So many things are need to be keep in mind when we are doing work with the larger animal models. So they are high cost, although they have more genetic similarity towards human, but the cost is relatively high. Then comes known human primate models, the organisms which are primates but other than humans, such as monkey, which has high maintenance cost. So as we are moving towards humans, which the organisms which are close to human, we are increasing their maintenance cost. So you can say there is a trade-off between the closeness towards the human genome versus their maintenance. So we decide on the basis of that which model organism we need to choose and which will be relevant to study the disease. So why animal models are needed? So human studies are limited due to ethical and practical constraints. We cannot directly study anything on the humans, so there are ethical concerns. So this animal model enables early drug evaluation before the clinical trials. So before initiating the clinical trials, before trying it on the patients, we first try it on the animal models. We generate mechanistic insights into the disease origin. We study that what is the disease mechanism, how it is happening. We model the rare and complex disorders. So there are around 550,000 studies in NCBI which use the animal models. So it is used across the cancer and neurodegeneration, cardiovascular and metabolic disorders. So all these disorders can be studied with the help of animal models. So let's see. So it shows the induced disease. So whenever we are studying the animal models, then they are not actually the patients, they are not suffering from any disorder. We induce the disease in them. So that could be pharmacologically induced. That means we are using some vaccine to directly put that particular antigens or anything related to that disease in the mus musculus, which is mouse. Or we induce the lessons in which we are making some injury in some part of the brain or some particular part of, means the region where we want to study. So the lessons can be induced. It could either be stress induced, so we can means keep model in such stressful environment which leads to the change in the behavior, or we can biologically induce it. So biologically we can supply something, such as in the case of rat, this thing is supplying biologically, we are inducing a disease state. Then comes spontaneous changes. So C. elegans could be used because in this we can see that what are the things which brings about the spontaneous change. Genetically modified animals such as zebrafish, which is also known as Danio radio. So in that we can easily do knockout knock-in things, so we can study them by making the genetic modifications. Negative models such as this SARS-CoV-2, it does not affect the pig, so they can act as a negative model. Then healthy animals, so we can see that healthy animal studies can also be done. We can compare them with the one who are infected, and then we can see that how the phenotype or the genotype is getting affected with respect to the disease state between the healthy animals and the diseased animals. Now there are certain characteristics of an ideal animal. So when we are deciding with which animal model to choose, then there are certain characteristics. If they are available in that model, then we will be able to choose among those organisms which will serve the purpose, which will serve our purpose. So there are certain points which, if are present, then they are considered to be the better animal models. So first thing is similar tissue distribution and route of infection as humans. Because we are studying the human disease, we are modeling the human disease in these organisms. So first thing which we want is that they should show similarity in the tissue distribution. They should not be completely different from the human tissues, as well as route of infection as humans. Short disease course, that means it should be completed with the quicker experiments. Reproducibility across trials, so we will not select any organism which is non-reproducible or which die off quickly. So how we'll study the disease for the longer time period? So it should be reproducible and easy to maintain, available in large numbers. So if the species are already limited, let's assume that I'm doing the research and I'm selecting a model which is already endangered species or which is about at the verge of getting extinct. So that is not a right choice of that animal model. So in that way it should be available in the large numbers. It should be cost effective and free of heavy regulations. So when we select the models which are very close to human, then there are many regulations, rules which are there. Or when we take the endangered or extinct, not extinct actually, but the endangered or endemic species, so in that case there are many rules and regulations which are available or which are levied on them, so we cannot use them. So we should select the ones which are cost effective and free of those regulations. Should show high pathological equivalence despite genomic differences. So even if they have the genomic differences such as zebrafish, Xenopus, Drosophila, although they have some genomic differences, but they should show high pathological equivalence. That means that particular virus or particular mutation must show the effect in them, must lead to a disease state in them as well. So zebrafish, it has transparent embryos for developmental biology. So whenever we want to study the developmental bio, that how the growth is taking place, we can use the zebrafish as a model organism. We can perform mutagenesis for genetics. We can see the results of CRISPR technology, or we can use it for the statistics for the cancer biology. If we talk about Xenopus, that it is the animal cap experiments for tissue induction, fate map of 32 cell embryo, dominant negative constructs, and morpholinos can be designed in Xenopus. Then mouse, it is excellent genetics with many banked mutants, physiology to test the organ functions, and tumor xenografts. If we talk about primates, then the organ physiology is similar to humans, so that is the most advantageous thing about primates. And if we talk about human cells such as cell lines or cell cultures, so transplantation of tumors and stem cells into the mice and cell culture can be done from the human cells. So these are some of the properties or points which were mentioned about each of these organisms, and these are the characteristics that when we are selecting the models, then what animal model will be more ideal, will be ideal. Now there is classification of animal models. So there are two categories which we have seen previously. So first is the rodents, where we have mice, rats, hamsters. They have short life cycle and they are small in size and quite cost effective. Other than rodents, we also have non-mammalian models such as zebrafish, flies, nematode, which are also low maintenance. Then comes large animals. So these are the large animal models such as pig, sheep, rabbit. They are close to human in many aspects. Then comes non-human primates, means they are primates but other than human, such as chimpanzee, baboon, gorilla. They are close to human genetically but limited by the ethics and cost. So there are many ethical rules, ethical grounds which exist, due to which we cannot directly perform experiments on these models. So there are certain limitations which exist, and others are the horses, apes, or special exotic animals for niche research. So we want to perform the niche level research where we are seeing that what is the habitat where they live, are the social animals. So that is niche research. We can use the exotic animals. Okay, so yeah, so as I discussed that there we need to make a trade-off, that there are certain animals which are easy to maintain but they lack uh, similarity to human. They are not very close to human, but in this end we have the animals which are close to human but they are relatively high cost and high maintenance. So we select among both of them those organisms which can serve the purpose. So then come mice as a model organism. So one by one we'll see that which organisms can act as the model organism. So first of all, starting with the mice. So its genome is about 85% identical to humans, and it has the orthologous genes which could easily be mapped and manipulated. So orthologous genes means the genes which are also present in human, the genes which arises from the same homologous gene from the same ancestor, but they have differentiated by the process of speciation. So different species, mouse is a different species, human is a different species. So the genes which are differentiated due to the speciation event, but they have same ancestor. So these are the ortholog genes. They could easily be mapped and manipulated. The advantages of using mice as a model organism is that it has a small size, so high throughput, cost efficient maintenance, and well developed genetic engineering tools. It is widely used in drug target validation, gene functional studies, and cancer research. So let's see all the points. So I have taken it from the Nature reviews. If you're interested, you can go to this site and you can read it in detail. So in this they have mentioned that if we talk about the life cycle of a mouse, then it is 4-day oestrus, 20-day gestation, 4 to 8 pups per litter, and 2 to 8 liters per female, 7 weeks to sexual maturity, and 2 year is their lifespan. So they have very short life cycle and they have better reproduction rate. We can perform reverse genetics, in which reverse genetic means that we are knocking out a gene knowing what would be the effect. Forward genetics would mean like we observe a phenotype and then we identify what is the gene. Here we know we are knocking out something and then we are seeing the change in phenotype. So this is reverse genetics. Then comes transgenics, where we are changing the genes, conditional expression such as Cre-Lox-P complex, where we have seen that we are conditionally knocking out some genes, because we have seen the reason for that also, that there are certain genes which are important for the functioning, and if we completely knock out them then the organism might die. So we perform conditional knockout, in which we only knock out that gene in certain tissues, whereas it is functional, its copy is functional in other tissues. So organism survives and we can see it conditionally in that particular tissue, that what is the influence of that knockout. Then inducible expression, which we can induce from externally. Then retroviral vectors and small interfering RNAs can be used, which can do the silencing of certain genes, certain RNAs. Then tools, genome sequence, embryonic stem cells, expression arrays, gene tap libraries, insertional vector libraries, back libraries. So there are different tools with the help of which we can perform these studies. Assisted reproduction, so we can also go for cryopreservation, in which we can preserve their embryos Embryo rederivation, we can obtain those embryos after cryopreservation. So in cryopreservation, we preserve them in low temperature, in cool environment. Then embryo rederivation, in vitro fertilization can be achieved. Intracytoplasmic sperm injections can be done for artificial fertilization methods, and cloning can also be done. So in that way we can do the assisted reproduction of, and there are different strains, so we can perform inbreeding, outbreeding, recombinant inbreeding, consomic and fluorescent. So these tests can be done. And if we talk about the genome, so it is a mammalian genome in which there are 22 chromosomes. Its size is 2615 MB, total 28,000 genes, and 99% of genes have a direct human counterpart. So in this way, mouse can act as a better model organisms, because in human we have different genes, and we know that different disease are caused due to mutation in those genes. So if we have the same gene in mice, then we can easily study the effect of the mutation in those genes in the mice. If those genes are not present in mice, then we cannot study the influence of mutation in that gene. We cannot artificially create that environment. So that is the reason that whenever we select a model organism, then we keep this thing in mind, that it should share some level of similarity to human at the level of its genome. Now next are the rodent and porcine models. So when we say rodent, then it means rats, and porcine means pigs. So rats are used in the cardiovascular and behavioral research. And if we talk about the porcine models, then they have physiological similarity to humans, and they are used in the cardiovascular diseases, diabetes and cancer studies. The example is the porcine models mimic the human cancer progression better than mice. But the main thing, the main drawback of the porcine models is that they are of high maintenance. So that is one of the reason that we don't choose them over mice. But if we want to study the progression of cancer, then we may work with pigs. So now we'll see applications of the rodents. So rodents, numerous models of obesity and T2DM have been performed on rodents. They are widely distributed and reproduce quickly, and they are available in the large quantities, and they are relatively cheap. Metabolic phenotyping technology available, and cost effective and multi-parity. So these are some of the applications. Now some of the cons of this. So pancreatic islet architecture is distinct from the humans. Monogenic models are not representative of most human diseases. The way we carry weight is different. So these are some of the cons, and these are some of the pros. Similarly with pigs, their operative capacity is strong. Evaluation of medical devices, surgical techniques, biological biological artificial pancreas, bariatric surgery, these all things can be performed on the pigs first. We can use these surgical instruments, surgical techniques, and we can study them on pigs, and then we can see that whether these are safe enough to be performed on the human or not. So those evaluations can be done. Technology maturity, so gene modification technologies can be studied in them, and then we'll see that how it performs. But the disadvantages of using the porcine model is that they are costly and specialized facilities are required. They have long life cycle, cost more than small animals. So this is some kind of trade-off which we need to make. So if our aim is that we want to study certain things which cannot be studied in the rodents, then we go for the pig, but keeping these limitations in our mind. Now we will talk about the spontaneous versus induced models. So spontaneous model means genetic mutations appear naturally in colonies. They mimic real human disease events. That means spontaneously the mutations are occurring in those colonies. Then comes induced models. So induced models are created by chemicals, radiations, viral or genetic engineering. For example, ENU mutagenesis, retroviral transduction and CRISPR edits. So in induced models, we are artificially inducing certain mutations, whereas in spontaneous models, the genetic mutations appear naturally in those colonies. So this is the example of spontaneous model. Animal models of CRC, chemically induced models in which we have used DMH to chemically induce the changes. Then this is the example of genetic engineered model in which tissue specific genetic mutations have been performed. So this genetic engineering is done by CRISPR, TALEN or zinc finger nucleases. So all those genetic engineering methods have been used. It could either be syngenic models in which the mouse cell lines are inserted into the mouse, but of the same organism cell lines are used. Then comes tumor tissue. So from human, this is the xenograft model in which we obtain the tissues or the cell lines from the human and then we artificially put them into the immune deficient mice and then we study the disease. So in this specifically they have focused on the cervical cancer disease. So in that way, these are the some type of models in which spontaneous induced means that the way how we have brought about the genetic mutations or perturbations, are those naturally appeared or are those artificially induced. Now methods of animal model generation. So different methods such as chemical and radiation induced mutations, because whenever we are studying these model organisms, so our aim is that either we can see the effect of mutations or we can see the knockout effect. So all those studies come under the disease models. That's why we use the animal models. So artificially we can induce the mutations using the chemicals or radiations such as X-rays. We can also perform the retroviral infection in which we can use the RNA viruses and we can infect these model organisms so that they can undergo the genetic modifications in their embryos. We can also perform the DNA microinjection in which we will put our genes or things in the injection and we can inject them into the model organisms in which we can engineer their DNA into the fertilized embryos. Then CRISPR-Cas9 genome editing. So targeted gene knockouts, knock-ins and base edits can be performed using this genome editing tool, and it enables the creation of precise disease associated SNP models. Now from this article I have obtained this method for generation of transgenic animal models. So if you are interested, you can go through this paper. It mentions in detail that what are the different methods with which you can generate the transgenic animal models. So I'll give you a quick overview of this. So this one is retrovirus approach. So retrovirus means we are using the RNA viruses. So this is a female donor mice from where you have obtained the 8-cell stage embryo. Now you are using the retrovirus which are carrying the foreign gene and you are inserting all of this into the pregnant female. Now the offsprings will be screened for the foreign gene. So the offsprings in this pregnant female, they will be having this foreign gene, so we can study it. We can study the effect of this foreign gene. Then comes regular transgene approach. So in regular transgene, we have a female donor mice from where we have obtained the fertilized egg which also contains the male pronucleus, and we have injected the DNA of male pronucleus here. So in this we are fertilizing this egg. This egg is obtained from the female pronucleus of the male and then we make the fertilized egg. Now we inject these oocytes into the mouse embryo and oocytes are implanted into the mouse embryo, and then later on we can study that how the offsprings come out, whether they have some kind of modifications or some kind of changes or not. Then comes gene targeted approach. So in gene targeted approach we have the embryonic stem cells from the embryo, and then gene transfer to the embryonic stem cells in vitro is done. Then these embryonic stem cells, they are injected into the early mouse embryo and then embryo is implanted into the female mouse. So this is gene targeted approach in which we are transferring the gene directly into the embryonic stem cells. Then there is types of transgene. This is the germline transgene and this is the conditional transgene. So the characteristics of germline transgene is that it is continuous expression of transgene. The gene expression can be promoter specific. Whereas the conditional transgene characteristics are, it is tissue specific transgene expression because we have LOXP construct, and then we have the knockout of the transgene expression in specific tissues. So use of transgenic animals as the biomarker source. in neurodegenerative disease. So blood, CSF, tissue and urine, these samples can be obtained and then they can be used. uh, these can act as the biomarker source in the case of the neurodegenerative disease. So in detail, if you want to read, you can go through this article in which they have clearly mentioned that how we can generate them in detail. Then comes CRISPR in animal models. So Cas9 system allows efficient and rapid genetic engineering, and it enables the point mutations, biohomology dependent repair or base editors, knockouts of disease genes, knock-in of human genes and humanized models. So it is applied to the mice, rabbits, zebrafish and pigs. So now in the right side, if you can see, so I have shown that this is the generation of cell lines and animal models using the CRISPR technique. So human cell lines, iPSC isogenic controls, mouse derived cell lines can be taken, zebrafish and pigs, and now we are using the CRISPR knockout, in which we can either knock out the gene, we can use CRISPR knock-in which we can use the HDR donor template, with which after the double strand break we can insert the gene of interest. We can use CRISPR-I, in which we can inhibit certain gene, because in this case instead of nucleases we have the crab domain attached to our guide RNA. So in that way it inhibits certain genes. Then CRISPR activation, in which we have VP64 complex. So instead of nucleases we have VP64 attached, and it will activate the genes. So we can do the lentiviral delivery of the single guide RNA library. We can select for the phenotype of interest and then we can go for the next generation sequencing. So this is the genome-wide CRISPR library screening. So this is how the CRISPR can be used for the different animal models. We can generate certain cell lines and animal models. We can make certain changes in the gene and then we can sequence it to check. Now comes iPSC models, and we'll look into the stem cell basic first. So stem cells are unspecialized cells which are capable of self-renewal and differentiation into multiple cell types. So this is the embryonic stem cells which are totipotent. So totipotent means it can form all the cells of the body and extra embryonic membranes. So as when we were looking into the disease in the previous session, then we see that in ex vivo therapies we have seen that we obtain the stem cells of the patients, and then on those stem cells we make certain mutations, we add the mutated gene and then we put it back into the host. So these stem cells have the ability to give rise to all the cells of the body. That's why from the bone marrow transplant is done. So in bone marrow there is the embryonic stem cells which exist, which are the hematopoietic stem cells. So now totipotency, the ability to form all the cells of the body. So oocyte, sperm, morula and blastocyst. So they all comes under embryonic stem cells. Then comes inner mass cells. So inner mass cells, they are pluripotent in nature. That means they can form all the cells of the body, but not the extra embryonic membranes. So they can form all the cells of the body, they are pluripotent. So these are the adult stem cells. This is the embryonic stem cells. So in case of adult stem cells, these pluripotent cells can give rise to digestive tissues, nervous tissues, cardiac tissues, and these are further categorized as multipotent, and beneath it we will be having the unipotent. So that means they can form a specialized lineage of cells that give rise to a tissue or organ. So in cardiac tissue also we have multiple such cells belonging to this tissue, so they come under multipotent, that they can form the specialized lineage of cells which give rise to this particular tissue or this organ, which is heart. Okay, so here I have shown a table. So zygote is a totipotent, embryonic stem cells are the pluripotent, specifically this inner cell mass. Then germ layer stem cells are multipotent, lineage stem cells are oligopotent, tissue determined stem cells are bi or tripotent, and then comes terminal cells which are nullipotent. That means they cannot give rise to any other cells. They are specifically the last ones, such as skin cells. Okay, so here I have given the types with examples. So unipotent means single lineage, such as skin cells. Oligopotent means few cell types, for example, myeloid stem cells. So these myeloid stem cells can further give rise to different types of muscle cells, which could be striated, unstriated. So in that way, they are oligopotent. Then comes multipotent, which means multiple related lineages, such as hematopoietic stem cells, which give rise to different types of blood cells. So they come under multipotent. Then comes pluripotent, that means they can give rise to all the three germ layers, such as erythropoietic stem cells and iPSCs. And then totipotent means they can give rise to entire organisms plus placenta. So they are, zygote is the example of the totipotent cell. Okay, now let's see what are these iPSCs. So they are induced pluripotent stem cells. So somatic cells which are reprogrammed back to pluripotency. So here we have seen that which can give rise to all the three germ layers, and as you can see, these inner cell mass. So when we are converting some somatic cells back to the pluripotent cell, that is known as induced pluripotent stem cells. So created using the transcription factors, Yamanaka factors, and these factors are OCT4, SOX2, KLF4 and CMYC, which are OSKM cocktail. So O stands for OCT4, S stands for SOX2, K stands for KLF4 and M stands for CMYC. And these all are the Yamanaka factors which are created using, these are the transcription factors. So iPSCs mimic the embryonic stem cells but avoid the ethical issues. So the key benefit is that they are patient specific, they are autologous, that means obtained from the same individual, so hence they reduce the risk of immune rejection, and they are powerful for disease modeling and regenerative therapy. So it is an example that we have the adult fibroblast cells. We can reprogram them and convert them into iPSCs, that is immune-induced pluripotent stem cells. Now these fibroblasts can give rise to cardiomyocytes, adipocytes, dopaminergic neurons, neural cells, motor neurons, pancreatic beta cells, and hematopoietic progenitor cells. So we are reprogramming the fate of these cells. We are inducing the pluripotency into these cells with the help of this cocktail, which is Yamanaka factor, which is the transcription factors cocktail. So this type of induced pluripotency comes into the induced pluripotent stem cell category. Now what is the history of these? So it began in 1962. Gurdon cloned a frog using the somatic nuclei and proved the nuclei retain the full potential. In 1996, Dolly the sheep was cloned by the SCNT. In 2006, Shinya Yamanaka produced the first iPSC mouse. In 2007, human immuno-induced pluripotent stem cells produced by the Yamanaka and Thomson groups. And then in 2012, Nobel Prize was won by Gurdon and the Yamanaka for reprogramming the discoveries. So this is how the iPSCs were originated, starting with the 1962 till 2012. So there are certain years which are not mentioned here, but you can go through them. So I have attached a paper in which the entire history, this review, the entire historical timeline which shows the events which had led to the development of iPSC is mentioned. So you can read it in detail, that how these events progressed, how the iPSCs were developed. So now let's see the application of these induced pluripotent stem cells. So first application is gene correction. So we can repair the mutations in the patient cells using the CRISPR based correction. We can do disease modeling, in which we can study the mechanisms of genetic disorders in vitro. Drug testing, we can use the iPSC derived cells and use them for the screening drug efficacy and toxicity. We can use them for regenerative medicine. So transplantation of corrected iPSC derived cells can lead to the regenerative medicine, precision medicine because they are patient specific therapies, and dosage prediction. So from patients only we can collect their fibroblasts, we can reprogram them, we can make them pluripotent, and we can treat the person. So in that way we don't have to deal with the condition of the non-self and self inhibition by our body. So our body will not be suffering from it because it will be specific to the same person. So it is patient specific, or you can say it is autologous treatment. Okay, so conventional approaches that we have Yamanaka vectors. We can use the viruses to insert the gene of interest into it, and we can make the somatic cell into iPSC, which is induced pluripotent stem cell. Then comes nanotechnology based approach, in which we have plasmid, we have mRNA, we have small molecules. With the help of particles, we can insert them into these particles. Particles can act as the delivery agents, which will carry these plasmids, mRNA, small molecules, and deliver them into the somatic cells. And then we can convert these somatic cells into the induced pluripotent stem cells. And here are the applications. So organoid production, we can produce certain organoids, we can regenerate tissues, we can use them in anti-cancer immunotherapy, we can screen the drugs, and then we can bring about the genetic modifications using these pluripotent stem cells. Now if we combine the immuno-induced pluripotent stem cells and the CRISPR, then what we can do? So we can perform knockouts, we can study the function of gene, we can do knock-ins, in which we can insert the disease relevant mutation for modeling. We can do transcription modulation, in which we can use dCas9 fused with the activator repressor proteins. We can go for the genome wide screening, in which we can use large CRISPR libraries to identify the novel targets. The example is that iPSC is differentiated into neurons for studying the Parkinson's disease. So we can study the Parkinson's disease using these iPSCs. And here is another figure from this particular paper. So in this it's mentioned that we have the healthy individual and the affected individuals. So after reprogramming, we can convert those cells into the immuno-induced pluripotent stem cells. So these are wild type human induced pluripotent stem cells, and these are affected human induced pluripotent stem cells. We can go for the knockout, knock-in, transcriptional activation, transcriptional repression, genome wide screening. So these all things can be done with the help of CRISPR-Cas9 gene editing methods. And after that, when we will make the changes in the genes, then we can study the differentiation of the edited human induced pluripotent cells. So we can compare them, that how these genetic modifications leads to the differentiation phase. And then drug development and monitoring of protein dosage can be done. We can study that how much protein dosage will be sufficient to treat that disease. We can go for the screening of different drugs, we can screen the gene therapy, and we can also study the immune response strategies. Then comes the case studies in these induced pluripotent stem cells. So first case study is of the Parkinson's disease, LRRK2 mutation. So this is the gene which is mutated. So it is corrected in the induced pluripotent stem cells by the zinc finger nucleases. So this is one of the case study. Second is the tauopathies in FTD mapped mutations. So modeled using the iPSC derived neurons. So in this case, neurons are derived and then they are modeled to study these tauopathies. So tau is generally so in these disease, different proteins, they make the complex kind of a structure, which is like fibrils, or as you can say, the amyloid clusters are formed in the brain. So these tau factors, these are when they are combining together forming a cluster, that comes under the tauopathies. Then pulmonary alveolar proteinosis, CSF2RA mutation. So it is corrected with the help of talins, which are transcriptional activator like effector nucleases. It demonstrates the potential of iPSCs for the neurodegenerative lung and metabolic disease. So all these case studies are running, so we can see that how we can use these iPSCs for studying all these things, all these diseases. So here is an example of the taken from this particular article, in which this is the patient. He has keratinocytes, urine cells, and PBMCs. So it's a healthy, one is the patient, another one is the healthy subject. So in both of the directions we are doing cell reprogramming, in which we are converting these cells into the induced pluripotent cells. And now these induced pluripotent cells are, because these are pluripotent, so they can get differentiated into the different cell types. So both of them are getting differentiated, then we can transplant them back. But before this we can use the genome editing strategies to edit these iPSCs. So in this case we have edited it, and in this case we have not edited it. So after editing we have cured for this mutation or the changes, because in the patient, if we know that okay, so patient is suffering from this particular disorder and responsible, this Parkinson disease is due to LRRK2 gene, so I can treat that mutation using the gene editing strategy. Now I have changed it. Now with this gene, when the iPSCs, they are cured, so when they will get differentiated and then they can get transplanted into the patient. So what I can do is I can go for drug screening, I can go for disease modeling, I can do cell therapy, I can see tissue engineering, I can go for the biobanking. So I can store it in the biobank so that the person can later on use them whenever he get any disease. So these are different types of cells which can give rise to these are multipotent in the nature. So we can engineer the tissues, cell therapies, disease can be studied, so we can compare among both of them, that is there any defect in these cells which are produced. So in that way we can compare with the help of iPSCs, that means we are comparing the disease model states, that whether my therapy is working or not. Now we'll talk about the cancer models. So starting with the history of cancer modeling. So in 18th century there was a person named Percival Pott. He linked the soot exposure to scrotal cancer in chimney sweepers. So he observed that due to the chimney soot, so the small particles which are present in the smoke or which are passing through the chimney, so leftover particles in the air, the soot. So when these chimney sweepers which were cleaning the chimneys, so they were showing the scrotal cancer cases. So he observed that this soot exposure is responsible for this scrotal cancer. So in 1915, Yamajiva and Ichikawa, they induced cancer in rabbits using the coal tar. So from here he has linked that okay, this particular exposure is leading to this cancer. So 1915, these people also, they induced the cancer in rabbit by using the coal tar. In early 20th century, Rudolf Virchow proposed cancer promoting chemical factors, and these all findings led to the experimental cancer models in animals. So starting with 1951, establishment of the first human cancer cell line, which are HeLa. So HeLa is obtained from a lady named Henrietta Lacks, that's why the name of the cell line is HeLa, and it is the most oldest cell line. This is the first human cell line. And this, as we say cell lines, so these cell lines could divide uncontrollably. These are like the cancer cells. So they are cell lines which have the ability to grow indefinitely. Then in 1969, the description of the first colon cancer patient derived xenograft model. So in the previous slides we have seen that xenograft model means from one patient we collect some samples and we put artificially into the model organism. So that thing is xenograft. In 1971, multi-cell spheroid culture as a model of nodular carcinomas. So these multi-cell spheroid cultures were used as the model. In 1987, introduction of a genetically engineered mouse memory cancer model. So in this case, they have used genetic engineering methods. to make the cancer model from the mouse. So in mouse they have induced the cancer. In 1989, a human colon adenocarcinoma cell line is grown in the transcells. In 2010, development of a lung on chip microfluidic device. In 2011, generation of the first colon adenocarcinoma patient derived organoid. In 2012, 3D microfluidic model of tumor microenvironment on a chip. So these are not just talking about the animal models, but they are talking about the cancer modeling when we are studying the how the cancer is behaving, and we are simulating the tumor microenvironment on a chip. Then in 2014, air liquid interface primary gastrointestinal organoid culture. In 2018, organoid modeling of the tumor immune microenvironment. In 2019, multi-compartment tumor on a chip model of lymph node tumor bulk crosstalks. And in 2020, automated microfluidic platforms of tumor organoids. So this is the field where the people have looked into the cancer modeling, that how the cancer is progressing. So this is known as cancer modeling. This is the another excerpt which I obtained from this paper. So in which they are talking about that in 1926 how the in vitro Carroll discovered the Rouse sarcoma virus which could be grown in the embryonic chicken tissues. So when we were talking about the ideal model organisms, then we have said that we can only take those model organisms which can show that particular disease state, which can show the gene defects as that of human. So similarly, in this case, Carroll has discovered that these viruses, they could be grown in the embryonic chicken tissues. Similarly, as we have discussed, in vitro establishment of the first cell line HeLa, and in silico Armitage and Doll, they published the multi-stage theory of cancer initiation. In 1960s, in vitro versus in vivo, so 3D multicellular spheroids were compared to the tumor grown in animals. Then after 1970s, in silico emerged, in which emergence of ODE models, and Gettin by and Gawalinsky published a reaction diffusion mathematical model to study how cancer spreads. So these ODE models are like when we are doing it, the mathematically we are modeling it, that how the cancer is spreading, what is the equation which can tell you that how the cancer is progressing, means with respect to time. So these are those models which were studied. Then ex vivo, so preliminary ex vivo models began to be published. Then after 1990s, in vitro and ex vivo, so emergence of biodynamic and microfluidic models, in vivo expansion of cancer studies performed in rodents. And if we talk about in silico, then human genome was sequenced and further advancing the in silico potential. Then after 2000s, we will talk about in vitro, then development of soft lithography driven lab chips, microfluidic co-culture circuits, and lab on a brain microphysiological systems using the biomimetic fibrous non-woven microporous transport barrier interfaces. We talk about in silico, then emergence of omics, precision medicine applications and modeling systems using the artificial intelligence. If we talk about ex vivo, then emergence of PDX models, bioreactor systems and personalized medicine applications. And after 2010s, emergence of multi-model cancer supermodels. So I don't think in this course you need to go into the detail of each of them. This is just the history. But if you are curious and if you are interested, then you can go through this link and you can read this paper, that how it initiated, how it started, and how the people looked into the cancer modeling and what are the cancer models which exist. So now what is the need for these cancer models? So cancer genome accumulates point mutations, chromosomal translocations, gains and losses. Mouse models critical to mimic the cancer complexity. So they are used to study the tumor initiation, progression, metastasis and drug resistance mechanisms. So novel therapeutic approaches can be achieved. Okay, so now one by one I will go through it. So what is the need for these cancer models? So this is the ex vivo model and these are the in vivo models. When we say ex vivo model, that means outside the living host when we are doing it in the culture, because I have told you that the cancer modeling is like you're studying the cancer state, and now it could either be inside the um, means this petri plate, or it could be inside the living organism such as the animal model. Okay, so let's start with the in vivo model. So let's take a person. This is the model interchange, and we have the animal models such as PDX, zebrafish, the cam. Now the advantages of in vivo model is that animal physiology, they have cell heterogeneity, because if I am performing it on the culture, then they have a single homogeneous cell state. So in the case of in vivo models, we have different types of cells, so there is cell heterogeneity, and then we have the physiology, the animal body, we don't have to replicate the same conditions externally. Then metastasis, so there is a balance between the states. Then complex ECM, and then the disadvantages are the high cost, special infrastructure, low throughput, and limited screening, so which leads to the informed treatment. Now if we talk about the ex vivo models such as organ culture, in which we are culturing an organ, or organoid which is 3D, or monolayer which is 2D. So the advantages, it is low cost, less labor, they can be used for genetic screens, they are high throughput. But the disadvantages is that defined cell types, defined ECM, low heterogeneity, and they are known metastatic. So when we say metastasis means that if I am studying a cancer, then we all know that not all the cancers are benign. They are metastatic. They can transfer from one tissue to other tissue, and one organ to other organ. They can spread. So in case of the in vivo models, I can study that if my cancer is in this tissue, it can move to the other tissue in the body in the case of metastasis, metastatic cancer. But if I am studying the ex vivo models in the organ culture, then in this case I will not be able to model the metastatic event, because I am taking one particular type of cells. So it is non-metastatic, and its heterogeneity is also low. It is quite homogeneous, and the ECM is defined, and its cell type is also defined. So these are some of the limitations. So that's why we need the cancer models, because we can study all these things. So it's about these, and I'm also showing the types. So both the types are important depending on what is our desire, so we can select any model out of them based on our need. Now there are methods for cancer models. So different methods with which we can study the cancer state in different animal models. So first is the spontaneous mutations in mice colonies. So spontaneous means we can naturally bring about those mutations. Then comes chemical and radiation mutagenesis, in which we will use certain chemicals such as ENU, UV rays, X-rays to bring about those mutations in the genes. Then comes retroviral insertion. So insertional mutagenesis can be done. DNA microinjection. So engineered oncogenes and tumor suppressors can be supplied with the help of microinjection. Then CRISPR and TALENs. So these are used for the precise translocations and mutations. Okay, so here I am showing the model organisms, and then different treatments which can be used. So first one is microinjection. So in microinjection we can directly put the whatever is present in this sample, which could either be the CRISPR construct along with the guide RNA or some gene. So we can directly put it into the male pronucleus by the help of microinjection. We can also use the viruses to deliver these agents, these RNAs, into the cells. We can also use electroporation, where we will use the electric current, creating the pores in the cell membrane and then inserting something into it. We can also use the nuclear transfer. So in which we can use the we have the enucleated oocyte, and we can insert the somatic nucleus into this enucleated oocyte artificially. So this is how we can make the cancer models. We can induce the cancer state in these model organisms by using these different methods. Now if we talk about the mouse cancer models, so the advantages is that it has short life cycle, about 2.5 years, genetic engineering flexibility, you can easily go for the genetic engineering in the mouse, and high cancer incidence with aging. So it's similar to humans, that with aging the incidence of observing the cancer increases. But now limitations are that mice develop the sarcomas, lymphomas, and humans mostly carcinomas. So this is the limitation, that instead of observing the carcinomas we observe sarcomas or lymphomas. And scale and metabolism differences with humans. So these are some of the limitations of using mouse cancer models, although they are quite advantageous. Yeah, so this is the example. Okay, so this is the syngeneic mouse in which the spontaneous mutation has been done. Here is a human xenograft in which we have implanted from the patient into the human, so that patient was the human, and from human we have entered the early disease, we have implanted the early disease factors into this mouse. So this is human xenograft, which could either be subcutaneous or intraperitoneal implantation, so disseminated disease. And in this case it is the syngeneic mouse, and here we have used the RNA interference or inducible gene expression or multigenic mutations, tet reversible gene expressions, and we have genetically engineered this mice. Now all of them can be studied, we can do imaging of them, and we can go further, we can do the reporter assays, we can study the immune therapies, we can see the tumor initiating cells, and we can profile their metabolism, that how the cancer state leads to change in their metabolic states. So this is how we can use the mouse cancer models. Now comes Ewing's sarcoma model. So it is the aggressive childhood bone cancer. This Ewing is aggressive childhood bone cancer, and 90% of cases have T1122 translocation in this EWSR1 FLI1 fusion genes. So these genes, this is a fusion gene, fusion of EWSR1 and FLI1. So if there is this translocation in 90% of the cases, this leads to the Ewing sarcoma model, in which there is this is the primary tumor, the primary site from where the tumor has started. So this fusion acts as an oncogenic transcription factor, and it is modeled using the zinc finger nucleases, induce the precise translocation, CRISPR-Cas9, reproducible and efficient model generation. Okay, so in this it is showing you the primary tumor site and the site of metastasis. So as I told you that this primary site could be different from the site of metastasis. Cancer cells can move from one tissue to other tissues. So this is the primary tumor site, in which this is the cancer tissues, they are now entered into the bloodstream, and from blood they have circulated to the other tissue, and this is the site of metastasis. So they have entered here through blood, and from blood through skin they entered into this tissue. This is known as extravasation. This was intravasation and this was extravasation. Now they will colonize in this, and this is the site of metastasis, where the from the primary site where the tissues have now entered. So that is site of metastasis, and this process is known as metastasis. And how the metastasis is taking place? So it is taking place through the bloodstream. Bloodstream is helping for the movement of these cancer cells from one place to another. Okay, now we'll see how we can use the talens in the liver cancer. So target genes are APC and beta catenins. Talens induced gain of function mutations in beta catenins and loss of function mutations in APCs. So it created a isogenic HCC cell lines and mouse tumor models. So HCC is the hepatic cancer cells. So this is the liver cancer, and mosaic mutations mimic the natural cancer progression. Application is we can study the Wnt pathway dysregulation and mutation synergy, that how the mutation is leading to these changes in the pathway. So here it is taken an example from the mouse. So the liver is isolated, and in these liver talin mRNAs are used, which are generating the talin proteins leading to double strand break, and then there is reduction in the LPA reduction, and we can artificially induce certain talents into the cell. So we can either perform it ex vivo, or we can in vivo also insert the talents. Now what it does, so when Wnt pathway is on, it's attached to the frizzled receptor, which leads to the series of reactions in which beta catenin enters into the nucleus, and then this beta catenin along with these factors, it leads to the gene transcription. But on the other hand, if there is dysregulation of Wnt pathway, then it will not be able to bind to this frizzled receptor, and ultimately the beta catenin will get degraded due to with the help of proteasomal degradation, which is the ubiquitin mediated proteolysis. Now if there is no beta catenin, it won't be able to bind to these factors, so no gene transcription will take place. So this tells you how the Wnt pathway is getting dysregulated. So this dysregulation can be studied with the help of the talins. Okay, now the CRISPR in the cancer models. So with the help of CRISPR, it enables you to knock out or the knock in germline models, somatic genome editing models, and chromosomal translocation models, such as in the case of leukemia and sarcoma. It is used to engineer the patient relevant mutations at high frequency, and it is critical for the preclinical drug testing and the biomarker discovery. So in this case, these are the organoids or the cells. With the help of CRISPR, we can either introduce the mutations, we can do the induction of changes in the transcriptional regulation. So here 2 rounds of the CRISPR is done. Now validation of drug targets, multiplex genome editing, and others. So we are doing the genome editing using this CRISPR technology. So these bars, as you can see, so these are the edited gene copies which are inserted into these cells, and this is just a representational purposes, but we all know that each cell has a DNA in the nucleus. So there we are making these changes using the CRISPR technology. Now this figure tells you about the brain. So we can use the lentivirus or AAV for the delivery. For the lung also, these adenovirus and lentivirus can be used, and intranasal and intratracheal delivery can also work here. For pancreas, we can go for the lentivirus or plasmid, or we can also go for the local injection or electroporation. For the colon, we can use lentivirus, and for blood cancer, we can ex vivo do the microinjection for that. For the muscles, we can use adenovirus or plasmid electroporation. For breast, we can use the lentivirus local injection. For the liver, we can use the plasmid AAV or adenovirus intravenously. Okay, so you are interested to read in detail, then you can go through this paper in which they have in detail told that how the delivery can be done. But for now, our main focus was that how the CRISPR can be used in cancer models. So this is how, with the help of CRISPR, we can bring about the genetic modifications in the organoids or the cells, and then these modifications can later on help us to study different disease states, or different treatments can be designed with respect to them. So we ultimately enter these organoids or cells into the model organism, and then we can study that how the model organism behaves. So this will be referred to as the induced cancer state. So we are artificially inducing the cancer environment within the animal model. So the conclusions related to this are, if we talk about the features, so animal models, iPSC models, and cancer models. So first of all, what is the relevance? So animal models, these are the helpful for the whole body physiology. iPSC models are the patient specific cells, because from the same patient We are obtaining those somatic cells, we are reprogramming them, converted them into the pluripotent stem cells. In cancer models, this is the relevance is that when we want to study the tumor biology. Now, advantages of animal model is that it provides us in vivo studies. So we are studying it in the live organisms. So we have heterogeneity of cells and multiple other advantages. We can study the metastasis, and all those studies can be done in the animal models. Whereas in case of iPSC models, it is helpful for the personalized medicine. When we want to design the personalized medicine for a patient, so because we are obtaining the cells, somatic cells from the same patient, so whatever medicine which we design, they will be very specific to those patients only. So personalized medicine. And if we talk about the cancer models, then they are closest to the human cancer genetics. So that is the advantage of cancer models. Now let's talk about the limitations. So limitation of animal model is species differences. That means I want to study or model the disease state of human in mice. So we both are different species. So we might not be able to study the 100%. We cannot say that there is 100% equivalence of disease states in both those organisms. So there are some species differences which exist. If we talk about iPSC models, then they are expensive and they are time consuming, so they will take a lot of time. They are quite expensive. If we talk about cancer models, then not all the human cancers can be mimicked. So this is the limitation of cancer models. Okay, so let's talk about the future perspective. So genome editing advancements, base editors, prime editing. So these are like future perspective with respect to CRISPR-Cas9 in the therapy of cancer. So right now we mainly focus on the CRISPR instead of ZFNs, TALENs. ZFN TALENs are the primitive approaches which were previously used. Now their use is bit limited as compared to the CRISPR-Cas9 technology. So if we talk about the future, then this CRISPR-Cas9, it can be used for the genome editing advancements. We can go for base editors in which we can edit certain bases, such as we can bring about the mutation in certain base pairs. So that comes under the base editors. We can do the prime editing. We can, means more than one nucleotide, we can also edit the larger fragments. Then humanized animal models for better disease correlation. iPSC based organoids, which is the induced pluripotent stem cell based organoids for studying the complex diseases. So with the help of iPSCs, we can study the complex diseases. CRISPR in oncology. So precise modeling of cancer subtypes can be done with the help of CRISPR. Movement towards the precision and personalized therapies. So this is the current scope, what is going on. Now in this I have taken the figure from this particular paper. So CRISPR-Cas9 in cancer therapy. So what are the different things which are ongoing? So first of all, we can go for the diagnostics. So because in CRISPR-Cas9 we have the guide RNA which binds to the specific site on the host genome. So if that guide RNA belongs to the defective gene, and I know that this this particular sequence leads to this disease state, so what I can do, I can enter my CRISPR into my host genome, and if it binds, I can run certain assays. If it binds, then that means that there is some complementary strand existing in the host genome, so which will confirm that yes, the disease state can be confirmed. So that is like a diagnostic tool, where CRISPR-Cas9 is acting as a diagnostic tool. Then CAR T cell. So here also this T cell, antigen and tumor cell, so they all, this particular CAR T cell therapy can also be used with the help of CRISPR-Cas9. And then comes PD1, PDL1. So this is also this is the tumor cell, and these are the receptors PDL1, and here it's the antigen which is binding to this, means which is on the surface of the tumor cell. Now T cells are coming with PD1 and T cell receptor. These T cell receptors can bind to these antigens, and then it can lead to the treatment of the cancer. Similarly, combating the carcinogenic viruses such as HPV, HBV, HCV and EBV. And then animal modeling, so different animals can be used as the model organisms. So in all of them we can use the CRISPR-Cas9 to study the cancer state. For example, in this case, if an organism is having the lack of T cells, so in the previous session when we were talking about the SCID, in which T negative or B negative or NK negative states exist, so in all those cases we can make the changes in those genes which are responsible using the CRISPR-Cas9, and then we can model that how those cells interact with the tumor cells, whether they lead to change in the phenotype, whether they kill the tumor cells or not. So in that way we can design the cancer therapy using the CRISPR-Cas9 approaches. Okay, so this is all about the how the CRISPR-9 technique has uh, been used in the cancer therapy. Okay, so with this now we will start with few MCQs for today's session. So if you have any doubts regarding today's session, so you can comment them. Okay, so now first question is, which of the following is a characteristic of spontaneous mutations in animal models? Option A, induced by physical stimuli. Option B, occur naturally in animal colonies. Option C, result from genetic engineering. Option D, caused by chemical agents. So you need to tell that what is the characteristic of spontaneous mutations in animal models. So I'll give you some time and then we'll discuss the right answer. Okay, now let's see. So right answer is option B. Okay, now let's see the question again. So it was saying that which of the characteristic is of spontaneous mutation in animal models. So we've already looked into the spontaneous mutations which occur naturally. So that means first option is wrong because it says that it is induced by physical stimuli. So this is not right. Option B was occur naturally in animal colonies. So this is true. Option C was result from genetic engineering. So this is not right because the result from genetic engineering is again the induced mutations. Caused by chemical agents is also the induced mutations. So right answer is option B. Okay, let's move to next question. Next question is, which animal is considered a non-human primate in research classifications? Option A, mice. Option B, pig. Option C, rabbit. And option D is chimpanzee. So you need to tell which animal is non-human primate. Okay, now so let's see. So the right answer is chimpanzee. Because if we talk about mice, so they are not primates. They are mammals but not primates. Similarly, pig, they are also comes under the large animals. Rabbit is also the large animals but they are not primates. So the primate is chimpanzee. Now next question is, which of the following is a method for generating transgenic mice? Option A, natural breeding. Option B, chemical exposure. Option C, physical exercise. And option D is gene editing. So I'll give you some time to think and then we'll discuss the right answer. So now let's see. So what is the right answer? So the right answer is option D, which is gene editing. So in this case, they are asking the method for generating the transgenic mice. So with natural breeding, we cannot generate the transgenic mice. With the chemical exposure also, transgenic means we are adding some genes from externally, from external source, which will be different from what gene is normally present. So that is transgenic. So which will not be obtained from the natural breeding process or the chemical exposure or from the physical exercise. So gene editing is the only right option available. Now let's move to the next question, which is in what year did Jenish and Mintz create the first ever mice with transgenes? Option A 1981, option B 1974, option C 2013, option D 1990. So you need to tell that in which year both these scientists create the first ever mice with transgenes. Okay, now let's see. So it's all right if you don't know the right year, because this is all about the dates. So in 1974, this two scientists, Jenish and Mintz, they created the first ever mice with the transgenes. So it's in 1974. Now let's move to the next question. Next question is, what is the primary gene editing tool mentioned in the lecture for creating the isogenic cell lines with specific genetic mutations? So option A is zinc, option B is NHEJ, option C is CRISPR-Cas9, and option D is HDR. So you need to tell which one is the primary gene editing tool. So you need to tell that what is the primary gene editing tool which was mentioned for creating the isogenic cell lines with specific genetic mutations. Okay, so now let's see. So the right answer is CRISPR-Cas9, because in this session we were looking into that how the different gene editing tools are used. So mainly CRISPR-Cas9 is used to create the isogenic cell lines. So before moving further, let's see what are the isogenic cell lines. So as the name suggests, isogenic. So that means the cell populations that are genetically identical. They are iso. Iso means genetically identical for specific intentional genetic difference. So there is only some specific genetic difference which exists, which could either be a mutation in one single gene or means which we are artificially inducing. So that is one, just in one single gene we are making the change, but otherwise it belongs to the same organism. So they just differ at the targeted gene or the mutation of interest. Otherwise they are the same genic cell lines. So that's why it is because of this it makes them the powerful tool to study the effect of single genetic change without background genetic variability. That means we are just making the change at the single site, but the entire background of that gene is completely constant, it's fixed. So let's suppose you have a normal human stem cell line, and by using the CRISPR-Cas9 you introduced a mutation in one of the gene which was linked to a disease. So now you have one original wild type line with no mutation and the other one is the mutant isogenic line with only that mutation which we induce with CRISPR-Cas9. So if we compare them, then it will reveal that what is the direct impact of this mutation on the behavior of cell without any interference from the other genetic differences. Because if we mutate multiple such genes, then how we will know that which gene is bringing about this effect. So that's why in the case of isogenic line, we are only mutating a specific gene and fixing everything else, so that if there is any change which is coming, that will only be due to that particular mutation. Okay, now let's move to next question. So next question is which stem cell type has the narrowest differentiation capability? Option A totipotent, option B pluripotent, option C multipotent, and option D is unipotent. So you need to tell which has narrowest differentiation capability. Okay, now let's see the right answer. So in this case, the right answer is option D, that is unipotent. So in this question they have asked you that which has the narrowest differentiation capability. That means which cannot differentiate into multiple such types. So we all know that these stem cells, they have the unique ability that they can renew themselves and they can differentiate into multiple other cell types. So when we begin with the totipotent, so totipotent are the cells which can give rise to any of the body cells, and especially they can also means all the cell types including the placenta. So example of the totipotent stem cells are zygote and very early embryos in their first few divisions. So the fun fact is that a single totipotent stem cell can theoretically create an entire organism. So if you have a single totipotent cell, you can create the entire organism. Now if we talk about the pluripotent, then these are the cells which um, such as adult cells like skin, blood, which can reprogram, and so sorry, in the case of pluripotent stem cells, they can easily become any cell types in the body, but in the case of totipotent, they can also make the placenta, but they cannot make placenta. So they can become any cells in the body except the placenta, and they can be used for the disease modeling and developmental biology and regenerative medicine. An example of the pluripotent cell is the early stage embryos such as blastocyst. Okay, let's talk about the multipotent stem cells. So multipotent can only become a limited range of cell types. For example, if we take the example of bone marrow or the skin or the gut, so the cells, they can give rise to only limited range of cell types. So for example, if your gut lining, so your the gut lining which we have, so it replaces itself every 3 to 5 days. So because they are multipotent cells, so they keep on replacing it in the intestinal crypts. So the application is that they are used in the bone marrow transplants in the case of leukemia or blood disorders. So the part which the bone marrow comes under multipotent stem cell. Then comes unipotent. So these are very or they, and sometimes they're also referred to as nullipotent. That means they cannot further give rise to any other cell types. They are just within themselves only. So we can also say that these produce only one specific type of cells, but they still have ability to self-renew them. That means they can divide and make more of themselves. So examples could be muscle stem cells. So they only give rise to muscle fibers. Or if we talk about the skin stem cells, then they can only generate the keratinocytes or skin cells. Although they are restricted to one lineage, but their self-renewable make them different from the fully differentiated cells. Like a skin cell which cannot divide indefinitely, but these cells, these skin stem cells which comes under unipotent, they can renew them. So that is the main difference between the fully differentiated versus unipotent stem cells. So I hope it is clear till now. Okay, now one more fun fact which I want to tell you is that these in these stem cells, they are also referred to as the blank slate cells of the biology, because they are like blank slates. They can give rise to all the cells, so that's why they are also referred to as blank slate cells. Okay, so let's move to next question. So next question is, what type of genetic alteration is most commonly associated with Ewing's sarcoma? Option A, chromosomal translocation. Option B, gene amplification. Option C, point mutation. And option D is gene deletion. So you need to tell that what alteration is associated with Ewing's sarcoma. Okay, so now let's see. So what is the right answer? So right answer is option A, chromosomal translocation. So now when we were talking about Ewing sarcoma, then I have shown you the image of a child. So it is a rare aggressive bone and soft tissue cancer, and it is most common in the children, teens and young adults, and it is characterized by this chromosomal translocation, and mainly the T1122 or Q24Q12. It creates the EWSR1FLI1 fusion gene, which is an abnormal transcription factor which drives this tumor growth. And this fusion is found in 85% of the cases. And this particular Ewing sarcoma, it mostly occurs in the long bones. For example, femur, tibia, humerus, pelvis, ribs. And it can also occur in the soft tissues, which is also known as extraosseous Ewing sarcoma. So whenever somebody is suffering from this Ewing sarcoma, so the symptoms are like localized pain and swelling, and it can also make you feel that you have a sports injury or infection at first. So that's why many times it gets unrecognized. So it is mostly for its diagnosis. We go for imaging or biopsy with the genetic testing for this EWSR1 fusion gene where the translocation has happened. And for the treatment, most people go for the chemotherapy or surgeries or some aggressive systemic therapies, but there is a high risk of metastasis, because we have seen that in this case primarily it's initiated at the level of legs, but then it goes to the other side. So it can go to the lungs or other sites, which are the secondary sites. It can get metastasized. So keep that thing in mind. And there is 70% survival in the localized disease, whereas if it get metastasized, then there is 30% chance of survival. Okay, let's move to the next question. So next question is, which of the following cell types can differentiate into all the cell types including extra embryonic tissues? Option A totipotent, option B pluripotent, option C multipotent, and option D is unipotent. So you need to tell to differentiate into all the cell types including the extra embryonic tissues. Okay, so let's see. So the right answer is totipotent, and it's quite straightforward because we have already discussed that totipotent are the ones which can give rise to all the cell types including the extra embryonic tissues and the placenta. Whereas the pluripotent, it can give rise to all the cell types but not the extra embryonic tissues. Multipotent is limited towards a particular cell types, and then comes unipotent, which has the ability to self renew, but they can only give rise to one particular cell. Whereas multipotent can give rise to one particular cell lineage. Okay, so next question is, which of the following mutations leads to an increased expression of 4R tau associated with the frontotemporal dementia? Option A P301S mutation, that means proline at site 301 is changing with serine. Option B mapped IVS 10 plus 16 mutation. Option C LRR K2 G2019 9S mutation, and option D is BRCA1 mutation. So what do you think would be the right answer? I'll give you some time to think and then we'll discuss. Okay, so now let's see. So the right answer is option B, which is mapped IVS 10 plus 16 mutations. So before talking about all these options, let's talk about that what is this 4R tau and what it is meaning in frontotemporal dementia. So this tau, it is the microtubule associated protein and it is found in the neurons. It is encoded by the mapped gene. So this is the gene which encodes the tau protein and it is associated with the microtubule and it is present on the chromosome number 17. So its main work, this tau's main work is to stabilize the microtubules which are like the tracks which help to transport the nutrients. So I hope you might be aware of the structure of microtubules. So they act as the tracks from which the nutrients vesicles, organelles, they all transport inside the nerve cells. So the function of tau is that it stabilizes those microtubules, the axonal transport which help to keep the intracellular traffic running smoothly, and they have also roles in the synaptic activity and brain plasticity. So there are different isoforms of this tau protein. So first one is 3R and second one is 4R. So R simply means repeat. Generally there are 6 isoforms in human brain of this mapped gene, and this 3R tau means there are 3 repeats, 4R tau means there are 4 repeats. And if we talk about the normal adult human brain, then it has 1:1 ratio of 3R tau and 4R tau. And if there is imbalance, like if we increase the expression of 4R tau, then it is linked to the neurodegenerative disorders, and in this case it is frontotemporal dementia. So now let's look into these options one by one. So in first option they are saying that P301S mutation. So as we know that this tau is in the mapped gene, and the effect is that this causes the abnormal tau aggregation and dysfunction, due to which it is linked to frontotemporal dementia. But this mutation does not specifically lead to the increase of 4R tau isoforms. Instead, this mutation can alter the tau functions, it can promote aggregation, but there is no known study that it can increase expression of this 4R tau. So in that way it is not the right answer. According to this, it says that this mapped IVS 10+16 mutation. So here IVS means intervening sequence 10. So the location is of this gene is on the intron 10. So the effect is that splicing of this exon 10, it increases the inclusion of exon 10, which results in the overproduction of the 4R tau repeats compared to 3R. So it's strongly associated with the frontotemporal dementia. So this particular mutation will lead to the increase of inclusion of exon 10, which will lead to the overproduction of 4R tau. That's why this option is right. Now next option was LRRK2G2019S mutation. So now we know that this LRRK2 is the leucine-rich repeat kinase 2. So effect is that gain of function mutation, it increases the kinase activity, and it is most common genetic cause of Parkinson disease. But in the case of frontotemporal dementia, this mutation is not responsible, and it has no direct association with this 4R tau. So this option is also not right. But in case of Parkinson disease, this might be the right answer because it will increase the kinase activity. Now next is BRCA1 mutation. So this is the DNA repair gene, and it causes the defective DNA repair, which is linked to the breast cancer and ovarian cancers. So it is also not related to the increased expression of 4R tau. So in that way, right answer is option B, which is mapped IVS 10+16 mutation. Okay, let's move to the next question. So next question is, who was the pioneer in observing the link between chimney sweep and cancer? Option A, Rudolf Virchow. Option B, Percival Pott. Option C, Katsusaburo Yamagiwa. And option D is Koichi Ichikawa. So this we discussed during the initial slides. Okay, now let's see that what is the right answer. So the right answer is option B, which is Percival Pott. Now let's look into the other options one by one. So if we begin with the Rudolf Virchow, we start with the Rudolf Virchow. Then he was the German physician, and who was also known as the father of modern pathology. So he developed the cell theory of disease. So in that way, he is not linked to the chimney sweep and cancer. Whereas if we talk about the Percival Pott, then he was the English surgeon. In 1775, he was the one who described that the scrotal cancer in chimney sweeps, means the sweepers who were cleaning the chimneys, so they were showing the scrotal cancer because they were exposed to the soot. So he was the one who established the link, Percival Pott. If we talk about the Katsusugaru Yamajiwa, so he was the Japanese pathologist in 1915 who produced the first experimental cancer by applying the coal tar to rabbit ears. So after the Percival Pott, he was the one who used the coal tar, and then he means experimentally brought about the cancer in the rabbit by exposing its ear to the coal tar. If we talk about Koichi Ichikawa, then he was the Japanese researcher who collaborated with the Yamagiwa in inducing the experimental cancer using the coal tar. So right answer is option B. Now let's move to the next question. So next question is, in designing the animal disease models, which aspect is emphasized most in the small rodent studies? Option A, ecological behavior analysis. Option B, natural mutation occurrence in colonies. Option C, seasonal breeding adaptation. Option D, plant animal genetic interactions. So what do you think was the aspect which is emphasized in the small rodent studies? Okay, so now let's see. So right answer is option B, which is natural mutation occurrence in colonies. So let's see all the options. Option A says the ecological behavior analysis. So it focuses on studying the animal interactions in their natural habitat. So it is not the main emphasis in the small rodent studies. Whereas if we talk about natural mutation occurrence in colonies, then yes, spontaneous or engineered mutations are inbred into the rodent colonies, so they are crucial for creating the reliable disease models. So yes, this aspect is emphasized. Option C was seasonal breeding adaptation. So this was relevant for the wild animals, but if we talk about lab rodents, then they are kept under the controlled breeding cycles. So this is not the priority in the disease modeling. If we talk about plant animal genetic interactions, so they are important if we want to study the ecological or agricultural studies, but not typically if we want to emphasize on the animal disease models. So the right answer is option B, which is natural mutation occurrence in the colonies. Let's move to the next question. So from 2018 to 2020, how did the number of mouse genes with phenotype annotation MP change? Option A, declined significantly. Option B, remained unchanged. Option C, showed consistent increase. And option D, completely disappeared. So what do you think has happened with the mouse genes with phenotypic annotations? Okay, now let's see the right answer. So right answer is option C, that is short consistent increase. So option A says declined significantly, so this is incorrect because research in mouse genetic has been expanding from 2018 to 2020. it's not shrinking. So declination is not the right answer. Option B, remained unchanged. So this is also incorrect because the annotation databases are continuously getting updated. Now the C option was showed consistent increase. So this is correct because there are some large scale projects which are running, like International Mouse Phenotyping Consortium, which is IMPC. It steadily added more annotated mouse genes each year. So number of genes with the phenotype annotations are increasing. That is consistently increasing. Whereas last option was completely disappeared. So this is also incorrect because phenotype annotations are core to mouse functional genomics. So they do not vanish. They remain. Okay, so now let's move to the next question. So next question is, which delivery approach has been used for the decades and is considered the most reliable for generating the transgenic animals? Option A, blastocyst microinjection. Option B, pronuclear injection. Option C, intracytoplasmic sperm injection, which is ICSI MGT. Option D is testis mediated gene transfer, which is TMGT. So you need to tell which delivery approach has been used for decades and reliable for generating the transgenic animals. Okay, so now let's see the right answer. So the right answer is option B, which is the pronuclear injection. Okay, so now this, in the pronuclear injection, the DNA is injected directly into the male pronucleus of the fertilized egg, and it has been used since 1980s. This is the first and the most reliable classical method for generating the transgenic animals. Now let's look at the other options. So first option is blastocyst microinjection. So it involves injecting the embryonic stem cells into the blastocyst, and it is useful for the knockout or knock-in mice, but not in the classic methods for transgenics. Similarly, if we talk about the option C, which was intracytoplasmic sperm injections. So in this case, sperms carrying the foreign DNA is injected into the oocytes. It is useful, but it is new, not the standard decade-old approach. Okay, if we talk about option D, which is the testis mediated gene transfer, then in this case, DNA is introduced into the testicular germ cells to produce the transgenic offsprings. So it is experimental and it is less reliable than pronuclear injection. So in that way, pronuclear injection is the approach which has been used for the decades and also reliable. So let's move to next question. Next question is, what distinguishes the gonad approach from other in vivo embryo gene delivery methods? Option A, require the viral integration into the genomes. Option B, eliminates the ex vivo embryo handling and microinjection. Option C, relies heavily on blastocyst injections. And option D, requires the repeated liposomal transfections. So what do you think would be the right answer? Okay, so now let's see. So the right answer is option B. That means eliminates the ex vivo embryo handling and microinjection. So we need to distinguish the gonad approach from other in vivo embryo delivery methods. So in this option, if we talk about the gonad, so that is the genome editing by oviductal nucleic acid delivery. So this method allows the nucleic acid or CRISPR reagents to be introduced directly into the embryos inside this oviduct. So this avoids the need for removing the embryos from the mother, culturing them and doing the micro manipulation injections. So that's why it eliminates the ex vivo embryo handling and microinjection. Okay, so option A was requires the viral integration into the gametes. So this is incorrect because gonads does not use the viral vectors and it uses direct CRISPR-Cas9 and nucleic acid delivery. Then if we talk about option C, so it relies heavily on blastocyst injections. So this is also incorrect because that's the embryonic stem cells or blastocyst injection method and not the gonad. If we talk about option D, requires the repeated liposomal transfection. So this is also incorrect because gonad uses the electroporation in situ and not the liposomal transfections. So the right answer is option B, eliminates the ex vivo embryo handling and the microinjection. Now let's move to next question. So next question is, in iPSC based neurological research, what is the benefit of creating the isogenic control lines? Option A, faster tumor progression studies. Option B, direct comparison between the mutated and the corrected lines. Option C, more rapid pluripotency induction. And option D is reduced need for the transcription factors. Okay, so now let's see. So the right answer is option B, direct comparison between mutated and corrected cell lines. So previously also we have talked about the isogenic control lines. Isogenic means everything else is same to that of the wild type, but the change is only at that one single gene. So previously if it was mutated, then now we have corrected it. So in that way it is easy to make the comparison between the mutated and corrected lines. And fast tumor progression study is more rapid, so all these options are not right and this one is the right answer. Now let's move to next question. So next question is, which genome editing tool is commonly combined with patient derived human immuno-induced pluripotent stem cells to screen the novel therapeutic molecules? Option A, CRISPR-Cas9. Option B, karyotyping. Option C, Southern blotting, and option D is DNA fingerprinting. So this is straightforward because the second word is the hint in itself. Okay, so let's see. So right answer is option A, which is CRISPR-Cas9, and we already know that how we can use this. But the other options were karyotyping, in which we look into all the chromosomes which are present. So this is not a genome editing tool. If we talk about Southern blotting, so then this is also not the right tool because in this we just move the DNA and we will just hybridize the DNA with the DNA band. So this is for the separation of DNA on the basis of their size. So this is also not the right answer. Then DNA fingerprinting, so this is also not the right answer. This is not a genome editing tool. So CRISPR-Cas9 is the only genome editing tool which is mentioned and is the right one. Now let's move to next question. So next question is, what is the primary aim of reprogramming patient somatic cells into the induced pluripotent stem cells for disease research? Option A, production of genetically identical animal clones. Option B, creation of lineage specific cells for disease modeling. Option C, analysis of ancient evolutionary variants. And option D is mapping of global DNA methylation arrays. So what do you think is the primary aim of these iPSCs for disease research? Okay, so now let's see the right answer. So right answer is option B, that is creation of lineage specific cells for disease modeling. So the advantage of this is the main advantage, because we have seen that in this case the patient from whom the specific cells are obtained, they will be specific to that particular cell lineage. So in that way our body will not be there is less chances of rejection of those cells, less chances or they will not get rejected. If we talk about production of genetically identical animal clones, so this was not the aim, and this is not what we want to do. Analysis of ancient evolutionary variant, this is also not true. Mapping of global DNA methylation, this is also not true. So the right answer is option B. Now let's move to next question. So to reveal disease specific mechanisms in induced pluripotent stem cell derived models, what factor is most essential? Option A, precise phenotypic characterization of derived cells. Option B, the patient's chronological age at biopsy. Option C, the number of colonies generated. And option D, the speed of dedifferentiation in culture. So what do you think will help you to reveal the disease specific mechanisms in iPSC derived models? Okay, now let's see. So the right answer is option A, which is precise phenotypic characterization of derived cells. So it's straightforward because we look further what is the phenotypic characterizations, because this will help us to know the disease specific mechanisms in these models. So these phenotypic characterizations is most essential factor. The chronological age at the biopsy has nothing to do with the iPSC derived models and to revealing the disease specific mechanism. Similarly, the number of colonies generated is also has nothing to do. Speed of dedifferentiation has nothing to do. So right answer is option A, which is the most essential factor. Now next question is, what chromosomal abnormality is a hallmark of Ewing's sarcoma? Option A, T1418 Q3221. Option B, T1122 Q2412. Option C, T922 Q34Q11. Option D, T821 and Q2222. So this we already have discussed in the previous question also. I have told you that what were the genomic changes in the chromosomal rearrangement in the fusion gene product of this Ewing sarcoma. Okay, so now let's see what is the right answer. So right answer is option B, that is T1122 and Q24Q12. So this particular chromosomal translocation is the responsible for this Ewing sarcoma because this leads to the fusion gene product. Now next question is, in TALEN mediated hepatocellular carcinoma studies, which gene showed the low efficiency of targeted editing due to polyploidy? Option A is APC, option B is beta-catenin, option C is HER2 and option D is RAS. Okay, so now let's see the right answer. So right answer is option A, which is APC. So APC is the gene which showed the lower efficiency of targeted editing due to its polyploidy. So this is the right answer, and with this we are in the end of this today's session. So we'll meet in the next session. If you have any doubts, then you can comment your doubts in the below the video in the YouTube, or you can ask your doubts in the discussion forums. And in the next session we'll look into the week 10 topics and we'll see the details. So thank you very much. I am ending the session here.